I thought it would be helpful if I gave you this lecture also on video so you can come back and you can review it as we go. So we already looked at some of this, but I think we'll just sort of start fresh and talk about chemical transmission and neurotransmitters. So when we talk about any of this, what we're looking at is a field called neurochemistry, when we're just focusing on what are the components of the nervous system in terms of the chemicals, the biochemicals that are being made. Neuropharmacology is also a field that studies the neurochemistry, but also looks at the compounds that could be man-made that can affect the nervous system. When we talk about things outside the bodies, we're talking about things which are, at least that are applied to the nervous system or to part of the body. We say that they're exogenous. So exo means from without, and gen is what's their origin. So the origin is from outside the body. And we've looked at the exogenous substances for, millennia, taking a look at what we take into the body and how that affects us. Very often we look at this, and we can talk about this too a little bit later. We'll look at the different types of drugs that are out there, some of the psychoactive drugs, and see what that means in terms of these exogenous substances and how they affect the nervous system. So the other term is endogenous. And endogenous just means that it's occurring naturally within the body. And when we're talking about endogenous ligands, very often we're talking about neurotransmitters, but there's other endogenous ligands that are that some of you are going to be researching, and we're going to see those too. So those can be neurohormones, neuropeptides. So there's different divisions of this idea of what's a neurotransmitter and what's a ligand. If it's a neurotransmitter, we know that there are certain things that they all have in common, and that's that they're something that is made by a neuron, and it's in the presynaptic terminal in those vesicles. So it's synthesized in the neuron, in the presynaptic neuron, and it's going to be released when an action potential reaches the terminal, and that's meaning that we're going to have this calcium channel opening, and it's going to activate those vesicles. The receptors will exist for this substance. If we consider it a neurotransmitter, we have to have receptors on the postsynaptic neuron, and then those are going to react or respond when that presynaptic substance engages them. And one of the ways to study that is if something is released by a neuron, but the receptors on the postsynaptic side are blocked, that tells us that the postsynaptic neuron doesn't have any response. That tells us that that particular substance is expressly designed by the body to activate those receptors. So this is, again, just a synthesis of all the things we looked at in class. So we know that neurotransmitters are synthesized in the neuron. They're found in the presynaptic terminal, but not just found there, but that they're in high concentration. They're released in response to calcium. They have, and that calcium release is dependent on this depolarization, right? So you know that because you remember that the saltatory conduction is basically that depolarizing that occurs all the way down the neuron during an action potential. And that at the end of the line, we have voltage gates for calcium, which are then also depolarized and open and allow calcium to come in. There are receptors on the postsynaptic cell. And when this is applied, if you isolate this substance and you apply it exogenously to a receptor on the other side, it's going to mimic that action that we normally see. So that's another way to test. And there has to be something that will terminate its actions. So now we're going to talk more in more particulars about neurotransmitters. Many of you have already chosen one to do your research on, so this is going to be sort of a broad overview. And there's different types depending on their chemistry, how they're designed. So amines have, you see the word amine, and that kind of fits to, at least with two of these, right? We see acetylcholine, and we see dopamine, and then we see another one called serotonin, which is sometimes called 5-hydroxytryptamine, which is, it's a sort of abbreviated name. And all of these are considered amines. And you may remember amines, at least that term, from looking at amino acids. But so the amine part of that would have nitrogen, right, and hydrogen. And we'll see the structure of this, or you will see, some of you, the structure of this as you're doing your research. There are some neurotransmitters that are simply amino acids. Here's the two biggies right here, GABA and glutamate. So remember your structure of an amino acid. You would see a central carbon, you'd see an amine group on one side, a carboxylic acid group on the other, and then some type of a functional group, R group, that would establish what GABA was versus what glutamate is. We have peptides. Okay, now remember what a peptide is, polypeptides. So we're talking about all pretty much the same types of molecules here. Peptides, though, are usually a little bit bigger, remember, because we have peptide bonds. So amino acids joined by these peptide bonds. So we're going to see those. And then interestingly, very interestingly, there's a group of them that are actually gases. And these two, nitric oxide and carbon dioxide, can both act as neurotransmitters. So here's just a chart that sort of shows that and it elaborates a little bit more on a couple of these. So if we look at acetylcholine, it's an amine. It's actually called a quaternary amine. And then there's monoamine. So this, you can see the word for in this, right? Quaternary. So we'll look at this structure. And then the monoamines, just mono meaning one, are these. And these are all related. So we're going to see how they're synthesized. Catecholamines include norepinephrine, epinephrine, and dopamine. And then another monoamine that is under this category, indolamines. Here's that serotonin with the 5-hydroxytryptamine right here. I may have misspoken and said tryptophan. It's tryptamine if I did. My apologies. So here's the abbreviation, 5-HT. And then there's melatonin. That's under that same category. Amino acids, we have GABA. And here you see the full name of GABA, which is gamma aminobutyric acid, but much easier to remember GABA. Glutamate, a cousin of glutamate, glycine, and histamine. Okay, these are all under that amino acid category. Neuropeptides, we're going to see things such as the opioids. Now you might say, what's that? That sounds like a drug that we're taking, but we have endogenous opioids and receptors for them. So those are the enkephalins, endorphins you've probably heard of, and the dynorphins. Some other neuropeptides exist, and there's oxytocin, substance P, you can see CCK, vasopressin, neuropeptide Y, and some hypothalamic releasing hormones. In other words, ones that the hypothalamus produces and will initiate a release of some other substances into the bloodstream. And then there's our gases. Oh, and this gas, as you see here, carbon monoxide. Neurotransmitters can't be neurotransmitters if they don't transmit something. And so they have to act on receptors. And receptors, as you remember, are proteins that are going to be embedded in membranes of all neurons. So you saw when we looked at just the standard neuron, we said, well, here we have ligand-gated receptors, we have voltage-gated ion channels, which are in some sense considered not exactly receptors, but they're still embedded in the neuron. But now we're going to focus on those ligand, the ligand receptors, or the ones that would be on the dendritic end. There are two main types. One is ionotropic, so ion you see here, and something that is ionotropic is something that is going to respond by opening up a channel for ions. So that's the response. They're ionotropic, and their properties are that they're super fast. So the neurotransmitter binds, changes the shape of the channel opens, the ion comes in, and then it closes. Quick on, quick off. But there are other types, and this one here, metabotropic, we just started talking about the other day. So these are slower ones, and when the ligand binds to a metabotropic receptor, it doesn't directly open a channel, because what happens is that binding will cause a series of other things to happen within the cell. And remember the G proteins are within the cell. They're on the intracellular side, and they're connected to the membrane to this metabotropic receptor. So we'll see these in a picture coming up. What you will see in your research is that when we look at receptors for particular neurotransmitters, there are things called receptor subtypes, which means that neurotransmitter may bind to different types, sort of subtypes of that receptor, and you'll see different responses. Here's the picture we looked at, or at least I think we got to this the other day. So you know that here's the presynaptic terminal for this presynaptic neuron. It must have had an action potential. This must have, calcium came in, activated the vesicle. The vesicle went to the terminal, to the membrane. Exocytosis, here's the neurotransmitter. It's coming across, binds. Now notice here, on this receptor side, we have binding occurring on two sides of this receptor, and that's opening the ion channel, and then the ion flows in. So if this was an ion such as sodium, what we get is a little wavelet of excitation right here. You get an EPSP, excitatory post-synaptic potential. Here's the other type of receptor, though, and that's this one right here. Notices there's no channel directly associated to this receptor, but instead, as the ligand or neurotransmitter binds here, it's going to activate this thing we call a G protein. Now, this G protein right here, I would call trimeric, meaning, remember, MER means like units. So trimeric means that we have three individual units as a part of this G protein, an alpha, a beta, and a gamma. And what you would normally see in order to activate this G protein, when the G protein is not activated, it's got a molecule called GDP associated. So that's like when it's on idle. But when this happens, this neurotransmitter binds, what it causes is that the GDP, or guanine diphosphate, gets kicked out and is replaced by GTP, guanine triphosphate. That activates it and instigates this activity. So notice that there's two units here. It's not showing the third part of this, this trimeric G protein. But some of these are going to activate DNA. They'll go in and actually activate genes to make something like some type of protein. And other parts of this can actually then go on and open an ion channel. So there's a bit of a delay, and this is a slower acting receptor, but it has more consequences. There's more things that happen in response. Neurotransmitters are ligands, right? So that's what we already know that. But when we start looking at neurochemistry and we start seeing what can happen, what we'll see is that there are drugs, so exogenous compounds, that can act to activate a receptor even when the neurotransmitter has not been released. And that is called an agonist. So agonist is going to initiate normal effects of a receptor. So that just means it's going to act essentially as the ligand. So if you were to be given a drug that was an agonist for a certain part of your nervous system, it would initiate the response that you'd expect, a normal response. You could also receive a drug in some cases, and we'll see some of these when we look at disease treatments, medical treatments, and you could receive an antagonist. So you know these from literature, an agonist and antagonist. An antagonist is something that blocks. Now, this third option, this inverse agonist, is one that it does initiate a response, but the response is opposite of what the normal ligand would do. So I'm not sure we'll see too many cases when we talk about this in class, but just so you are familiar with it, you have sort of these three options. Something that's an agonist or an antagonist or an inverse agonist, all of these are considered competitive ligands because they go to the receptor and they do something there. So they're going directly there. There's also some type that are non-competitive. And a non-competitive ligand is going to do something, it's going to bind, but it's not going to bind to the normal site that the neurotransmitter would. It'll bind to some other site. And that's going to affect the activity of that particular neuron. It could affect it in a couple of different ways. These are called neuromodulators because they're modulating activity. Here's an example. So here's an ionotropic receptor. Notice there's a channel that's associated with it, but it's closed. Here's the neurotransmitter. It's binding. And as it binds, we open the channel. Now it's open. So here we have something that would be an agonist because it's a drug. It was made outside the body. In other words, it's exogenous. It bound, causes the same effect as the normal neurotransmitter. The antagonist has bound. In this case, it results in the receptor not opening. So it opposes, it's in opposition to what the normal one would do. And then this one here, notice that there's a different site that this one is binding on. So this is a non-competitive antagonist. So it binds to some other site. And even if the normal neurotransmitter binds, it can't activate. So that's a non-competitive because it doesn't affect the original site of the neurotransmitter, but it stops its activity. And I think I'll stop right there.